In this video, we'll be going through enzyme inhibitors, which is a part of the biological molecules topic. If you haven't seen my previous enzyme video, click here and give that a watch. There are two types of reversible inhibitors that you need to be aware of: competitive and non-competitive inhibitors. These inhibitors prevent substrates from binding, therefore less enzyme substrate complexes can form, hence the amount of enzyme activity is reduced or stopped. Competitive inhibitors compete with enzyme active sites, as the inhibitor has a similar shape to the substrate. As the inhibitor is directly blocking the active site, it is competing with the substrate for the active site. Whereas the non-competitive inhibitor does not compete for the enzyme's active site, the inhibitor binds to an alternative site called the allosteric site, which alters the active site's shape, which prevents the substrate from binding. In your exam, it is important that you are able to differentiate between the two and are able to apply this to exam questions, as they may give you a scenario of different types of inhibitors and ask you to identify what type of inhibitor it could be and why. So you may be wondering, why are inhibitors even needed if they just slow down enzymes' activities? As with anything, inhibitors are there to act as regulators in metabolic pathways. The graph shows you the rate of reaction against substrate concentration. This line shows you the rate without an inhibitor. This line is with competitive inhibitors, and this line is with non-competitive inhibitors. So with competitive inhibitors, the initial rate of reaction is lower than without inhibitors, as the competitive inhibitors are occupying some of the enzyme's active sites. As the substrate concentration increases, they outcompete the competitive inhibitors. As more enzyme substrate complexes can form, the rate of reaction increases. Overall, the end amount of product that can form is the same as without inhibitors, but just takes longer. With non-competitive inhibitors, the initial rate of reaction is also lower than without inhibitors. As the non-competitive inhibitors bind to the allosteric site on the enzyme and change the active site shape, the substrate can no longer bind to the active site, hence less enzyme substrate complexes can form, therefore reducing the rate of reaction. Regardless of increasing the substrate concentration, the enzymes are still inactive as the inhibitors have changed the active site shape, so a plateau is reached. The enzymes that are still able to work will continue to form enzyme substrate complexes and continue at the rate of reaction. Overall, the end amount of product formed would be less than without inhibitors. As I said before, if you haven't seen the enzymes video, go give that a watch. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.